So the research that I'm most interested in is exploring the ecology of microbial communities that are responsible for transforming nitrogen in ways that contribute to losses of nitrogen from agroecosystems. So I'm going to talk about a couple of different lines of research that we are pursuing in order to explore the microbial populations that are responsible for nitrogen transformations and the factors that control their activity. In doing so, I hope that we can micromanage nitrogen losses. So I'm going to talk about some of our NREC funded work that is looking into managing the microbiome, the soil microbiome that's involved with nutrient losses. I'm going to talk about some currently funded work and then some preliminary work we've done in other directions. So first off, I'll start out talking about how microbes get a lot of bad press. So you can see that they're famous for infectious diseases, plant diseases, contamination of food. But I also want to point out that we live in a microbial world. We're surrounded by a microbial milieu. The soil has a lot of microbes. We interact with a lot of microbes. And so it's not so easy to get away from microbes. And we've definitely evolved in this microbial world. And it's important to understand that the ways in which we partner with microbial communities in order to promote environmental health. So there's a lot of benefits that are derived from microbial communities too. So even just in the soil, there's a lot of different microbial populations, most of which do not cause disease or any sort of detrimental effects. So I just wanted to give a quick overview of the microbial processes that occur in aquatic and soil ecosystems. So microbes are responsible for transforming carbon, of course, all these different carbon transformations. They fix and respire carbon. They are responsible for nearly every transformation in the nitrogen cycle. So microbes are really important for that. They can take up phosphorus, and they can transform sulfur, and they can talk to each other as well. So in all those processes, all those functions, they are influencing environmental quality. The other thing I want to point out before I leave this slide is that different microbes carry out different functions. So when we are interested in microbial ecology and the composition of microbial communities, it's because we understand that different microbial populations have different functions. So for example, in this nitrogen cycle, if these pink microbes are missing, then we lose that critical transformation of nitrogen gas to plant-available nitrogen. So that only those microbes in my cartoon community can fix nitrogen. So composition matters for ecosystem function and the ecosystem services that microbes provide. Okay, so I mentioned that microbes can control most all of the nitrogen transformations. So the major ones are here. We've got mineralization, nitrogen fixation, nitrification, and denitrification. And different microbial populations are responsible for all of those transformations. The other thing to know is that, I'm sure that most of you are familiar with this, a couple of these transformations are not so desirable in agroecosystems. So nitrification transforms plant-available ammonium that will be retained in the ecosystem. Nitrification transforms that to nitrate, which can be easily leached out of our agroecosystems. And then denitrification transforms nitrate to greenhouse gases or to atmospheric nitrogen, but it's lost from the ecosystem and represents a decline in soil fertility as well. Okay, so we're interested in understanding the ecological drivers, the factors that influence the microbial populations that are responsible for each of these transformations, and then understanding how those transformations are affected by our management decisions. And this has consequences for environmental quality, of course, because transformations of nitrogen that lead to losses from ecosystems can occur throughout the growing region of the Mississippi River Basin. And all of that nitrogen is collected, all that nitrate is collected in coastal ecosystems such as the Gulf of Mexico. And leading to eutrophication, hypoxia in the Gulf of Mexico dead zone. And this occurs not just in our Gulf of Mexico, but really anywhere that anthropogenic nutrient inputs affect coastal ecosystem water quality. Okay, so with all of that as background, I want to talk about how microbial ecology can help with this. So I'm an environmental scientist and also a community ecologist who happens to study microbes. So I study patterns in space and time of microbial communities in order to understand the factors that control the microbial communities and their activities. So I look for these patterns. I try to I then relate those patterns to potential ecological drivers or management factors that are influencing the microbial community. I'm particularly interested in the consequences for any of the nitrogen cycling processes, particularly soil fertility and nutrient retention in this case. And of those ecological drivers, I want to find out which of those are ones that we could manipulate through management decisions. So basically, I'm approaching nutrient retention as an applied microbial ecology problem. Okay, so can we manage the soil microbiome? So from a microbial ecology perspective, managing the soil microbiome requires that we identify the controls on microbial communities and their functions, and then identify ways that we can influence those drivers of microbial communities through agronomic management. I'm also interested in exploring how we can alter microbial communities through crop breeding decisions too. And through this, we want to develop a framework by which we could predict and predictively manage microbial communities. So what is it specifically that we want to manage? So of course we want to enhance soil fertility, so and provide nutrients to plants. And we can do that. Microbes can contribute to this by fixing nitrogen or by mineralizing nitrogen from soil organic matter. And in addition to that, we'd at least like to leave nitrogen in, if we're going to fertilize crops, we at least want to leave the nitrogen in the field. So we'd like to reduce denitrification and in terms of losing nitrogen and also reducing greenhouse gas production. We'd like to reduce nitrification as well, and we'd like to improve dissimilatory nitrate reduction to ammonium, which is going to be the focus of the first part of my talk here. So dissimilatory nitrate reduction to ammonium, DNRA, is a pathway to reduce reverse nitrification. So reverse the transformation of ammonium to nitrate. So DNRA transforms nitrate back to ammonium. And this is a way that we transform that mobile nitrate back to immobile ammonium. And the way that we do this, so I'm going to just quickly talk about the methods that we use to do this too. So then, the microbial nitrogen cycle is really nice because the genes for looking at the microbial populations involved with nitrogen cycling, the genes that are diagnostic for these functional groups, are relatively well conserved across our soil microbes. So we can use those genes, and I've got our diagnostic genes designated here. We can use those genes to characterize the microbial populations that are specifically involved with these different transformations. So we do that through DNA sequencing and quantitative PCR of those diagnostic functional genes. We compare the community composition of the functional groups or the abundance of those functional groups with our rates of nitrogen transformations, and then we evaluate that in the context of other experimental factors that we included in our observations. Okay, so this is an overview of the terrestrial nitrogen cycle. And just to remind you of what it is that we're trying to manage. Okay, so we've got ammonium, nitrification transforms that ammonium to nitrate, and then the process that I'm going to be talking about first here is DNRA, which transforms that nitrate back to ammonium. And so we're viewing this as a potential mechanism for nitrogen retention. Because that nitrate is easily lost, the ammonium is not so easily lost. Okay, so why haven't you heard of DNRA before? So DNRA is only recently recognized, and the people who are studying DNRA mostly have been studying it in coastal ecosystems and salt marshes. So it was thought to be really more important in aquatic habitats. it was also thought that denitrification was more favorable under a lot of different conditions too. So basically, no one ever thought to look in soil for DNRA, even though this seems like a really good idea for nitrogen retention. So the prevailing model was that this activity was confined to saturated soils rather than upland soils like we have in our agroecosystems. Okay, but recently there's been some shifting evidence that suggested that DNRA might be more widespread than just coastal ecosystems. So DNRA microbes were detected using high-throughput sequencing approaches that have become available in the last decade or so. And then also, DNRA was found to be occurring under unexpected oxygen conditions, so where there's a little bit more oxygen than would be favorable for denitrification, which requires anaerobic conditions. Okay, so this is some data from a collaborator of mine in California ecosystems showing that DNRA rates are positively correlated with soil nitrate, and that DNRA is happening in a lot of different soils. So you can see that these are all upland soil. Well, most of them are upland soils, and there's plenty of DNRA happening in these upland soils. So this led us to ask, is it also happening in agroecosystems? So remember that DNRA positively correlated with nitrate in that previous slide. And here in Illinois, we have lots of ecosystems that have plenty of nitrate in them, and soils that would really benefit from DNRA. So NREC funded our work that has the goal of understanding the extent to which DNRA happens in agroecosystems and the potential controls on DNRA. So what are the different management factors or environmental factors that influence the rates of DNRA as they are occurring in the agroecosystems? So we went to agricultural fields, a lot of different kinds of fields in the Urbana area, and we characterized the abundance and diversity of microbes that were responsible for dissimilatory nitrate reduction to ammonium, and then correlated those rates with different management practices. So the different approaches that we used stable isotope methods to measure the rates of dissimilatory nitrate reduction to ammonium, and then we used DNA sequencing and quantitative PCR to measure the composition, the diversity, and the abundance of the microbes that were responsible for these factors. Okay, so this is a complicated slide, so I'll walk you through it just a little bit. So each one of these points on these plots represents a microbial community, a soil sample, and we measured the microbial community. The distance on each one of these plots represents the dissimilarity in microbial community composition. So communities, points that are very far apart represent microbial communities that are very different in composition. Points that are close together represent microbes that are very similar in composition. And we look at these sorts of plots and we look for patterns in community similarity that tells us something about the factors that control microbial communities. So in these plots, you can see that that there are differences in microbial communities based on the type of cropping system that they were sampled from. So we have ones that are in conventional corn-soy rotation and no-till, and then also where residue was added or not. Okay, so we can see that by the clustering of the points of different colors, we can see that those factors influence, those management factors influence the microbial communities in these ecosystems. So already we're seeing some differences. We're starting to discern some of the controls on microbial communities. Now, the similarity, so we also found, though, that although the microbial communities were different, the composition was different among those agroecosystems, we didn't really see much difference in the abundance across some of these systems. Now, some of them, we did see somewhat greater some differences, some minor differences in the abundance of DNRA, but not dramatic differences, but some variation among the cropping systems. Okay, but then looking at the activity of the actual rates of distillatory nitrate reduction to ammonium, we can see that DNRA exhibited this bimodal response. So we have higher rates of DNRA, there we go, at low moisture. So this is moisture, soil moisture on the x-axis, DNRA or denitrification rates, so DNRA on the top, denitrification rates on the bottom, on the y-axis. And we see that DNRA exhibits this bimodal response to soil moisture. We have higher rates of DNRA at lowish soil moistures. And then as we start having the soil moisture that is conducive to denitrification, where denitrification is winning, a little bit more denitrification here, that's where DNRA drops out. And then at even higher soil moisture conditions, those two processes can co-occur. So this is really, really an interesting phenomenon that hadn't really previously been observed. So we've got DNRA happening at soil moisture and oxygen levels that are really too low to support denitrification, and so that's really great. We get a little bit more moisture where denitrification is winning, and then even more moisture where they are co-occurring. So upwards of about 21% soil moisture, we're starting to see more denitrification there. Okay, so our take-home points, or the summary of the results that we've generated to date, include that our agricultural soils around here in Illinois perform DNRA at meaningful rates that could represent a potential mechanism that we could enhance for soil nitrogen retention. Now, even though we have very different microbial communities across our different cropping systems, the DNRA gene abundance was relatively consistent. There was some variation among cropping systems, but not really dramatic differences among the different cropping systems. So, consistent across, relatively consistent in abundance. And we found that DNRA rates were controlled by soil nitrate, and I didn't really show those data, but we have some evidence for that too. But soil nitrate and soil moisture. So we're starting to discern some of the drivers on DNRA in our agroecosystems. Further work that we're going to do will be to evaluate these drivers through more controlled lab experiments, so we can kind of explore some of the controls on DNRA. We've done some of that work already. We'll be doing more. The other thing is we'd like to know specifically which microbes are active. So we know that the DNRA microbes are there. We know that they're active because DNRA is happening, but we don't know the identity specifically of the microbes who are actively carrying out DNRA. And understanding those which microbes specifically are carrying out DNRA might give us a little bit more fine-scale view of which of how we could control and manage this process. So we'd like to once we have the identity of the active DNRA microbes, then we can look and see how they're responding to different soil or nitrate conditions. Okay, I want to shift gears a little bit here. So DNRA work I shared was all about reversing nitrification, but it would be better if we could just reduce nitrification initially. So now there's already a precedent for altering below-ground nitrogen cycle to conserve nitrogen in ag soils. These are some of the commercial products that are available for this purpose. But I'd like to talk about the potential importance of plant microbiome interactions too. So in terms of reducing the potential for denitrification. So corn breeding programs tend to focus on above ground traits, and they have, they take place in conditions with high levels of nutrients, and a lot of the factors, a lot of the functions that these plants would have relied on microbes for are taken over by our agronomic management decisions. So we reduce competition, we provide nutrients for these plants. So basically these plants, modern crop plants, have a reduced reliance on microbiome functions. Now, we are interested in kind of revisiting this topic because we know that crop genotype affects the soil microbial community. Different plants will be colonized by different microbial communities. And so we'd like to understand the extent to which we can control microbial functions by choice of crop genotype. So we're also interested in understanding the extent to which we might have lost microbial functions throughout the process of crop breeding as well. So what got left on the editing room floor as we were selecting for above ground traits? So we went all the way back to ancient genomes, and we're comparing the domesticated, the microbiome of domesticated corn with the ancient lineage Teosinte. So again, this is another one of those figures where each point represents a soil microbial community collected from the roots of corn in blue, or Teosinte is green, and the soil microbiome that was started with in this experiment is over here in brown. So from that soil microbiome, each lineage, the modern maize or the Teosintes, recruited distinctly different microbial communities. So you can see there's two different clusters of the green and blue points. Distinctly different microbial communities were recruited from that same soil. So that was very exciting. Our next question was, does this matter for microbial function? Are these crop genotypes, are these different plants selecting the microbes based on the functions that those microbiomes produce? Okay, so we looked at nitrogen cycling functions because we're really interested in nitrogen retention. And the first thing we saw is that the denitrification is suppressed in Teosintes. So this is out in the field. These are actual field rates of denitrification. Corn and Teosinte, modern corn and Teosinte were planted in the same field, managed the same way, and denitrification was significantly suppressed in the Teosintes. We also found that nitrification rates were suppressed in the Teosinte. So again, these are receiving the same amount of nitrate. They're managed conventionally, but the Teosintes reduced denitrification. So up until this point, I actually was not familiar with biological inhibition of nitrification, but it turns out it's a thing. A lot of ancient lineages, or older lineages, wild lineages of related grasses are able to produce root exudates that inhibit nitrification. So there's a number of these known already. So this is the nitrification pathway on the bottom here. The root exudates act at different enzymes, act on different enzymes, inhibit the enzymes involved in those transformations. This hadn't previously been observed in corn, however. It's been observed in sorghum and rice and wild relatives of wheat and barley as well. But this phenomenon had not been observed previously in corn, probably because no one looked as far back as Teosinte. So from our initial results, we demonstrated that the maize microbiome has been changed through the process of domestication and crop improvement. And we've demonstrated that that has altered the nitrogen cycling functions of the root-associated microbiome. But in order to truly take advantage of that, we need to identify the plant genetic markers that control and improve the nitrogen retention. So to start doing that, we made use of some near-isogenic lines, some maize Teosinte near-isogenic lines to explore what potential Teosinte loci were responsible for the suppression of nitrification and denitrification. So these maize Teosinte near-isogenic lines, it's a cross between maize and Teosinte, back crossing to the B73 maize until we end up with a panel of near-isogenic lines that are mostly B73, each with a chunk of Teosinte genome in them. And we planted a set of those near-isogenic lines that represents the entire Teosinte genome distributed across a bunch of different near-isogenic lines. So we planted those out in the field again, and we replicated our previous results of suppressing nitrification. So again, we've got our B73 with plenty of nitrification and our Teosinte suppressing nitrification. Most of our near-isogenic lines had the B73 phenotype, which makes sense because they are mostly B73, but we were able to find 2 near-isogenic lines that exhibited the teosinte phenotype. What that means is that those near-isogenic lines have genetic loci that are responsible for suppressing nitrification. So now we've demonstrated that this phenotype, this suppression, this biological nitrification inhibition phenotype can be conferred onto modern maize. We also looked at the microbial communities associated with these near-isogenic lines, and we found that there was a correlation between how much nitrification we observed and how much colonization by nitrifiers was observed too. So our plants that had high nitrification rates had higher levels of colonization by nitrifiers. So we're also interested in denitrification, of course, too, and so again, we repeated our results showing suppression of denitrification by teosinte, and again, most of our near-isogenic lines had the higher denitrification rates that are typical of the modern maize. But we found 3 lineages, 3 genotypes that had the suppressed denitrification phenotype as well. Okay, so then our next steps are to map the specific teosinte loci to recruitment of the microbiome and suppression of those phenotypes. So we're using some bioinformatic approaches to, and some additional near-isogenic lines with overlapping teosinte regions to narrow down the genetic loci that are involved with suppression of denitrification. So ultimately, we are looking to identify the mechanism for suppression of nitrification and denitrification. We'd like to identify the root exudates that are responsible for these activities and the plant genetic markers that are responsible for generating those root exudates, and see if we can introgress those into modern maize lines while still maintaining high yields of modern maize. So with that, I'll take any questions.